Hello, my name is Blaudez and I'm presenting today my PhD project, which is the harnessing the native extracellular matrix for periodontal regeneration, where I'm being supervised by Prof. Sasho and Dr. Cedric. So first of all, the problem that we are trying to target is periodontitis. Periodontitis is a destructive inflammation that occurs around the periodontum. It usually starts by the formation of a biofilm, which then leads to an inflammation that becomes chronic and eventually will lead to the loss of the tissue that surround the teeth. The most common treatment is the guided tissue regeneration, which consists in cleaning the area, placing some bone particles in the defect to promote bone formation, and then place an obstructive membrane between the defect and the gum to prevent from gingival fibroblast infiltration. So the advantages of these techniques is that it allows good wound stabilization and spence maintenance, and it also allows for a selective cell repopulation. However, the limitation of such techniques is that it's usually quite unpredictable in terms of results, and it does not allow structural regeneration of the periodontum and therefore no functional regeneration. Tissue engineering is an interesting way to solve such problem because it aims at combining scaffold with cells and signaling molecule to restore some complex structures. So the aims of this study is to create an off-the-shelf biophysics scaffold that can allow a compartmentalized regeneration of both the alveolar bone and the periodontal ligament. And we have two hypotheses. The first one being that the osteoblast deposited ECM can promote the bone regeneration. And the second one is that the decellularized biophysics scaffold can promote periodontal regeneration. So the research plan for this study is, in the first place, to fabricate the scaffold. So we first printed with multi-electrowriting our meshes with predefined and already optimized structure for bone formation. And then those scaffolds were cultured in vitro with osteoblasts stimulated for ECM production, and they were decellularized after 1, 2, and 4 weeks of culture. We then thoroughly characterized in vitro our construct, both cellularized and decellularized. And we also performed a recellularization study to study what biological properties have the different matrices after the different time of cultures. We then assess the bone healing potential in vivo of the different matrices in a cardiac model. And we implanted both cellularized and decellularized scaffold to compare the performance. We then combine the best performing group for the bone regeneration with periodontal ligament cell sheet, and we decellularize the whole biophysics construct to prove that our technique is translatable. So when looking at the decellularization evaluation, so we could observe that we had a constant and homogeneous deposition of collagen, as you can see in the confocal images in green, and that was mineralizing over time, as you can see in the alizarin red staining. And when studying the cellular components of the different scaffolds, you can see in the confocal images, the blue, which is the nuclei, and the red, which is the cell fibers, have completely disappeared in the decellularized scaffolds. And we also confirmed this by a picogreen quantification showing that sufficient removal of DNA was obtained. We then recellularize our construct with human osteoblasts to see the behavior of these cells in vitro, along with a control. And what we observe is that all the ECM group allowed for a higher cellular attachment, and that the cell proliferation was high at 1 week, but it seemed to be impaired in 4-week ECM. We also ran an ALP activity study, ALP being a marker of early osteogenic differentiation, and we could see that a more mature matrix display a greater osteogenic activity. So we then studied the potential of these different matrices in vivo by comparing the different time of culture and also evaluating the performance of both cellularized and decellularized groups. We also implanted a pristine scaffold and also had an empty defect as controls. So when looking at the microCT, we could see that overall decellularized scaffold and cellularized ones perform very similarly. We also observed high vascularization in all the groups except in the empty defects. Overall, we had higher bone formation and a more mature tissue in both the 1-week groups. So we then selected our 1-week construct as our best performing group, and we combined it with a periodontal ligament cell sheet before decellularization. So we did some characterization in vitro to verify that our decellularization protocol could be used in this context as well. So when looking at both the confocal and the SEM images, we could see that we had efficient cellular removal, and we also had a good preservation of the extracellular matrix and the overall structure of our construct. So our decellularization protocol efficiently translated to our biphasic construct. As a conclusion, we could see that a short maturation of our construct only was successful for promoting bone regeneration, and that longer incubation resulted in a dense and obstructive matrix, which would eventually impair the cellular infiltration and probably remodeling of the matrix as well, and overall affected bone formation. So we then combine our 1-week bone compartment with a periodontal ligament cell sheet before the decellularization, and we generated an off-the-shelf biphasic construct with periodontal regenerative potential. I would like to thank the I-team, which is this amazing group of multicultural researchers with who I've been spending my last 3 years. And they've been helpful in a lot of aspects of my PhD and my everyday research. Thanks everyone, and thanks for your attention.